Okay, good morning, friends. Today I'm going to discuss about the general structure of chromosome and its types. See, we have different type of chromosomes are present in our body, and especially chromosome is a genetical material which are helping in the genetical heredity. Especially, I mean the genu means the genetical product which are present in the chromosome structure, especially in the genes which is located on the particular on the surface of chromosome, that place which is called as locus. Okay, so the chromosomes are also as called as hereditary vehicles, especially, and this type of chromosome was first discovered by half Mr. Scientist in the year of 1848. Okay, then chromosomes మొట్టమొదటిసారిగా means the chromosomes are first Tradencantia plant we can identify in especially the cells of pollen grains. Okay, that is especially the pollen grain mother cell we can identify. After that, Waldeyer he given a particular name, that is a chromosome, chrome, which means that is a color, and zo, which is nothing but that is a particular structure. Okay, that is a chromosome. Okay, colored structure. Now, after that, the chromosome theory was discovered by Sutton and Baveri in the year of 1902. Okay, see, uh, uh, Sutton and Baveri they given a particular theory of chromosome basis of heredity. Okay, then the chromosome proper structure we can identify it in the divisions. We have two different type of divisions in present in our botany, especially for example, I'm going to discuss about the stages of mitosis and meiosis. The mitosis which present four different type of stages are there. That is the first one, um, meta and anaphase and telophase and prophase. See, the proper structure of the chromosome which is clear visible in prophase, not in the telophase. Okay, now now I'll go with the general structure of the chromosome. Just wait. This is the common general structure of the chromosome. We can identify the chromosome which has a different type of parts are present. That is the first one that is a chromomere. Sorry, the first one chromatids. Okay, the second one shortylite. Then third one that is nothing but centromere, and fourth one that is nothing but sat. Okay, so these are the common parts which is present in chromosome. Now I'll explain about the chromosome number. The chromosome number is highly different plant to plant, even human beings, even animals. So now I'll explain about different type of numbers which is present in the plant. I'll go with Ziamis, which are having only 20 chromosomes, which is nothing but 10 pairs are present. Okay, the next one, onion means allium cifa, which present 16 chromosomes are present. Okay, so plants are mainly haploid or diploid. See, haploid is nothing but single chromosome is present. The diploid is nothing but that is 2 ends chromosome, and triploid cell is present, that is pen, the primary endosperm nucleus. This primary endosperm nucleus is which is located in the reaction of triple fusion process in angiosperms, especially the reproduction time. Okay, now the third and fourth one that is triploid, and fourth one that is tetraploid, which means 4n. We can observe the polyploid. Okay, so and next usually all developing plants and all developing plant bodies which are having two set of chromosomes, that is diploid in nature. Okay, next, this is called as genome. Fine. And now I'll give some examples, the chromosome numbers which is present in different type of plants. First, Elium which pairs 6, the number of chromosome only 16. And next, Batani, that is the Physum Sadevum, which are having 7 pairs, which means 14 chromosomes are present. Is it clear? Next, and Nicotia tabacum, that is Pogaku, which are having 24 pairs of chromosomes are present, that is nothing but 48 chromosomes are present. Oryza, that is only 12 chromosomes, 12 pairs of chromosomes are present. Indirectly, the total number of chromosomes are 24. Last but least, that is Goduma, Triticum vulgare, which are having 21 pairs of chromosome, that is 42 chromosomes are present. Okay, and finally, our human beings also different type of chromosomes are present. There is 23 chromosomes. The 23 pairs of chromosomes are present. That is especially the entire chromosome number is 46 in a human being. See, the chromosomes are mainly in our body. I am telling, especially the chromosome size, that is especially 0.25 μm in the fungi members, to 3.2, that is a trillium, that is the highest biggest chromosome which is present in trillium, up to 3 μm. Okay, in eukaryotic plants, the chromosomes are mainly two types, that is there is autochromosome and allochromosome. Autochromosome C means which is not participating in any sexual reproduction and which are having high number. Then allosomes, in our human body, autosomes and allosomes are there, and out of 46 and 44 are belongs to autosomes. Allosomes is nothing but only having one pair, that is two pairs, XX chromosome and XY chromosome. These are the sexual chromosome pair which is participating sexual differentiated are male and female. Okay, now the chromosomal structure. The chromosomal structure we can identify as different type of centromere and chatelet and telomere and chromonimata. Okay, so chromonimata, in telophase we can observe different type of chromonimata like air-like structure. Like this is the chromonimata, which can observe two different type of chromatins like heterochromatin and euchromatin. What is euchromatin and heterochromatin means? So euchromatin, the which can absorb color is little amount and which are having very active nature. Heterochromatin, that which can absorb color is more, the genes in the region of heterochromatin which is not that active when comparing euchromatin. Okay, so the chromonimata is first discovered Bejerenki scientist in the year of 1880, and he is discovered in the plant of Tridentcantia plant, especially the pollen mother cell. Okay, after that one more scientist is there, Wijdowski, in the year of 1912. He is also first proposed chromonima. Is it clear? And see, the chromonima is up to 60 angstrom length is present. Okay, this type of chromative, on the surface of the chromatin we can observe a bulbous structure. These are called as chromomeres. See, each chromomeres which present DNA plus histone proteins are present. These are help in the heredity, especially heredity. Okay, next, This type of chromo coils we have to identify, that is paranemic and plectonemic. What is paranemic is nothing but which are having very loosely arrangement, and plectonemic which are arranged as clumsy. Okay. Centromere. This is the primary construction which is present in the chromosome. We can identify it in the divisions, especially in the metasis. We can observe like a kinetochore and fibers which is present, and in the microscope we can observe in the time of metaphase the centromere, which is divided into two arms of a chromosome, especially based on centromere. The chromosomes are mainly four different types are present here. That is first one, if centromere is present, that is if centromere absent, that is called as acentric, and if one centromere is present, that type of chromosome is called as monocentric. If two centromeres are present, that is called as dicentric, and more than two, that is called as polycentric. Okay, so holocentric is centromere is not at all clear visible. Okay, so based on centromere location, again the chromosomes are classified into four types, that is metacentric and submetacentric and acrocentric and tetracentric. So metacentric means the chromosome arms are equal in size. The centromere is located in the central. Okay, this type of chromosome which is located like a V-shaped structure. The next one, submeta chromosome. The centromere move forward to some right side or left side, and arms are different size. One arm is big and one arm is very small in size, which is located as L shape. Okay, acrocentric. Okay, the chromosome which is located as J shape structure. Okay, and telo, and only one arm is present, and the centromere which is located in the tip of arm, this is looking like a I shaped structure. This uh, L and J and V and J structures we can identify in the anaphase. In the division in the particular metaphase, we can observe in the anaphase. Okay. Now, I'll go with uh, secondary construction. The secondary construction, this is a similar character with the primary construction, which and the secondary construction with metaphor of RNA, and which are helping in the RNA with metaphor of some genes, and also which are having 8S and 28S, and different type of secondary construction which are help in the nucleolus. This is the arising part of the nucleolus. Okay, that is a very important. Is it clear? Then finally, that is shortlet. Shortlet is a tip of the chromosome which is present in the lower, and this is looking like a round shape structure, and also shortlets, which means short, which means shine acid and thymonucleosine. Okay, this is thymonucleic acid especially, which is present in the DNA, but this short is diploid in condition. Okay, and finally that is a telomere. In the chromosome, the telomere, the tip of the chromosome which is called as telomere, that is first of all the telomere was discovered by Muller scientist in the year of 1938. Okay, and the telomere with metaphor of DNA and estrogen proteins which are help in the symmetry, means the chromosome which are help in the particular stability of the telomere, which are helping the symmetry and stability. Okay, if any changes which is occur, we can identify as the telomere. Okay, These are the general structure of the chromosome. Is it clear for you people? Just finally, I can explain outline. First of all, the general chromosome structure was discovered by world scientist. First of all, after that, Saturn and Bavary will go with chromosome theory was first discovered by Saturn and Bavary. The chromosome is the general structure which is present in the lower side of the cell, the nucleus, which are helping the genetical heredity, and the chromosomal size is very minute, then very longest chromosome up to 3.5 million. Next, the chromosomes are mainly two types, that is auto and allo chromosome. Auto chromosomes, it is called as vegetative chromosome, which is not participating in any reproductive. Allosomes are, that is the sexual chromosomes, which have two pairs in human beings, that is XX and XY chromosome. These are the male and female identified chromosomes especially. Okay, the next one, the chromosome structure, we can observe different type of part, that is chromonemata, and also primary construction and secondary construction in short and telomere. And see, the chromonemata, it is a hair-like structure which is present in the inner side of the chromosome, like this structure, and with colored and non-colored. Then colored, which is called as heterochromatin, very active, and euchromatin. Okay, so and chromatin, which can observe the bulbous structures, these are called as chromomeres, and the chromomeres with made up of DNA and histone proteins. Okay, the chromomeres which is present in the gene, a gene in the located part is called as that is locus. Okay, and next, primary construction, this is also called as centromere, which is divided into two parts, the centromere, the primary construction. And next one, we can observe the secondary construction. The primary construction, we can observe kinetochore and fiber bonds, especially these are which is present in the divisions, especially metaphase. And the secondary construction, this is a nucleolus organizing part, the secondary construction. Okay, and here there is no kinetophore and there is no spindle fibers are absent here in the second construction. Okay, next, this is the outer and inner chromonematous, and this is the outer structure, and shortlet is a tip of the chromosome structure, and telomere also, the telomere which are helping the symmetry, asymmetry, and also which are giving stability. And also the chromosome types, based on centromere number, the chromosomes types are classified into 4 to 5 different type of chromosomes. If centromere absent, that is belongs to acentric. If one centromere is present, that is monocentral. If two centromeres are present, that is called as dicentric. If more than two, that is called as polycentric. Okay, next I'll go with the centromere position. Based on 4 different type of chromosomes are present, that is telocentric and metacentric and submetacentric and acrocentric and telocentric. So metacentric, which means the centromere which is present in the central location, and arms are two equal in shape, and which is located like a V shape in the anaphase, while during division. Submetacentric, is nothing but somewhere the centromere is moved forward, and the arms are two different shapes, not at unequal size. One is big size and one is small. which is located like a L-shaped structure. Next, acrocentric, this is looking like a J-shaped structure, and telocentric, that is which is located like I shape, means the arm is only single arm is present here, a tip of arm which are having a proper centromere, means which is looking like a I shape in the face of one of it. This is the different type of chromosomes which has present like meta, submeta, acro and telos, including these. And also we have allo and autosomes, and that is especially human beings in our human body, which is having 46 chromosomes are present, which means 23 pairs. Out of 46, we have two pairs specialized chromosomes, that is allosomes, that is identified the male and female XX chromosome, and XY chromosome. The XX belongs to female, and XY chromosome that is belongs to male. This is the general structure of chromosome, and the introduction and structure, and also chromosome types based on centromere and number, and based on centromere position especially, and also vegetative and sexual chromosome types. What is the chromosome function is nothing but which are helping in the genetical heredity, and also which are help in the transfer of genetical material from one generation to another generation. These are the most important topic of chromosome structure. Is it clear? So this is the general structure you can observe. This is general structure, outline structure of the chromosome. You can identify it in under microscope like this structure. This is the outer and this is the inner structure of the chromonemata, and chromosome structure very clear visible like primary construction, secondary construction, even satellite and telomere. And here on the right side we can observe chromonematas like euchromat and heterochromatin. Okay, this is the types, and this is a clear visible euchromatin and heterochromatin, right? Is there any doubts you can ask me? And very important topic for you people, and these are the very important topic for MSc. Whatever you go with MSc, whatever you go with group 4th and group 2 and other, and the given from cell biology 4 to 5 questions, especially 4 to 5 questions is nothing but, for example, I'm telling you people who was first discovered by chromosome, that's Valde, and the chromosome theory was discovered by Saturn and Bafferi. Okay, what is the function of primary construction? And I'm telling not particular this topic, I'm telling entire cell biology. For example, who was proposed by nucleolus, that is a Fontana. Nucleus was discovered by Robert Hooke. The cell wall with made up of different type of lamellas. Before I can explain now, that is the primary lamella and secondary lamella and middle. So the middle lamella with made up of which products? That is very important, very hard in nature. That is the calcium and magnesium, which means the calcium carbonate and magnesium carbonate, hence which has hard in nature. Okay. Next, we go with DNA. For example, DNA. So, what is nucleotide and nucleoside? They are asking in MS question. Nucleotide, which means which has having 3 different type of parts, and nucleotide, which means which has having only 2 parts. Okay. Next, what is pyramids and purines? What is the ratio in between pyramids and purine in the RNA and DNA? What is main difference between RNA and DNA? These type of questions they can ask in the entrance exams. Okay, like MSc botany, whatever PhD entrance, and whatever group 4th and group 2 and other group 1. Okay, why I'm stressing cell biology is a very important, is nothing but they people given up to 5 questions to 6 questions especially. And here very important cell organelles are there like mitochondria and chlorophyll and plastids, and ER endoplasmic reticulum and lysosomes and peroxisomes and vacuole. These are the cell organelles, including cell divisions also present, that mitosis and meiosis. So what is mitosis, meiosis, and our work was proposed, and also which stages are present, like prophase and metaphase and anaphase and telophase. What is mitosis and meiosis differences? So these are all important topics. Okay. Okay, thank you, students. Thank you.